Santham Academy. Welcome to Digital Kaksha. To get updates on new videos, click the subscribe button and the bell icon.
Welcome back to the chapter Principles of Inheritance and Variation. In the last video, we have seen the Mendel's monohybrid cross. So there we have simply crossed the individuals just like the cross multiplication method. Now one more method is there to make the cross between two individuals, and that is called as the Punnett Square method or the checkers board method. So what is this Punnett Square or the checkers board method? This method was designed by the scientist Reginald C. Punnett. In this method, graphical and diagrammatic representation of crosses is being done and probability of the genotypes of the offsprings is being determined. Here the gametes, they are written on one side and the boxes are there in which the possible combinations are being put. So here you can see that this cross we have already done in the form of cross multiplication in the monohybrid cross, but here in the form of Punnett Square we can see that this side we have written at the corner the male and the female gamete. Now the male and female gamete will be capital T, and here the female will be small t and small t. Now we can make the combination like capital T, small t, capital T, small t, small t, capital T, small t, capital T. So we can see that we have got the same result what we have got in F1 generation. So all the offsprings or hybrids in F1 generation will be phenotypically tall and genotypically they will be heterozygous tall. So this Punnett Square method or the checkerboard method is simply a diagrammatic or graphical representation of the cross in square boxes in which we can see the probability of genotypes in the next generation. So this was about the Punnett Square which was discovered by the Reginald C. Punnett. Now we will move to the dihybrid cross by Mendel. You all know that what is a dihybrid cross? A dihybrid cross is a cross in which two individuals are crossed for two contrasting characters. In dihybrid cross, Mendel have crossed two different plants which bears two different characters, that means two genes and 4 alleles. So when we cross two plants taking two characters, 2 genes or 4 alleles into account or consideration, such type of cross is called as the dihybrid cross. In monohybrid cross, we took only one character, and that was the height of the plant. So one character was there, so 2 alleles were there, that is the tall and the dwarf, and we have crossed that. But here in dihybrid cross, we will be taking 2 characters at a time. So let's see what are the 2 characters we are going to take. Number 1 is the shape and number 2 is the color of the seed. So 2 characters we are taking or 2 genes we are considering, that is the shape and color of the seed. So the shape of the seed, the dominant shape is round and the dominant color is yellow. And when we go to the recessive trait, the recessive color is green and the recessive shape is wrinkled. So we have considered 2 characters or 2 genes, that is the shape and color of the seed. The shape, that is the round and the wrinkled. Round is the dominant one and wrinkled is the recessive one. while the color of the seed, that is yellow, is the dominant one and green is the recessive one. So here we have took the pure lines, that is pure round and yellow and pure wrinkled and green. So now these two plants will act as the parents. Now as they will give the gametes, you can see that as capital R and capital Y here will form the gamete, and here small r and small y will form the gamete. After that, these both gametes will combine to form the F1 generation. So this F1 generation will be having round and yellow seeds but in heterozygous condition. So here we can see the genotype: capital R, small r, capital Y and small y. The genotype here is capital R, small r, capital Y and small y, and the phenotype will here be heterozygous round and yellow seeds, as round and yellow are the dominant alleles. So they will be expressing themselves. Now we will move to the cross, that is the self cross. Now we have got the F1 generation. So in the F1 generation we have got all round and yellow seeds. Now we go for the self cross of F1 generation. So this F1 crossed with F1. So here again we will form the gametes. You can see that on a corner we have represented the male and the female, and here we have written the possible combination of the gametes. So here you can see R and Y, capital R, capital Y, capital R, small y, small r, capital Y and small r and small y. So the same gametes will be written on this side also. So here as it is self-crossed or it is self-pollination, the gametes will remain the same, like the same gametes or same combination of alleles is written on this side and the same combination is written on the other side. So we can see that when we cross them, we can get the variety of resultants or variety of offsprings. So we will perform this cross by the Punnett square. So as 4 gametes are there, so there will be 16 squares, and these 16 squares will be giving or representing the different genotypes. So now let us see how the crossing is being done. So here we can do the cross as this crossed with this. Now capital R, capital R, capital Y, capital Y. This will be the first combination. The second combination will be capital R, capital R, capital Y and small y. Again from this one, capital R, small r, capital Y and capital Y, the third one. And now the fourth, capital R, small r, capital Y and small y. So in this way we will be getting the combinations. You can see how easily we can cross with the help of a Punnett square. So simply you have to write the gametes on both the sides and cross them or put the 2 alleles together. 2 alleles on one side and the 2 alleles on the another side. So putting all the 4 alleles together, you will get the genotype. So in this way, after crossing this 16 combinations we will get. So let us see what is the phenotype of these 16 combinations. Number 1, you can see capital R, capital Y and capital Y. So here you can see capital R and capital Y. That means the dominant allele is there. So the dominant one is the round and the dominant color is yellow. So here the round and yellow seeds. Now the next, here also you can see capital R and capital Y. So here also the round and yellow seeds will be there. Here also you can see capital R and capital Y. So here also round and yellow. Here also round and yellow. In this way you can see that how many round and yellow seeds are there. Let's see and count how many round and yellow seeds are there. 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9 combinations of round and yellow seed. Okay. The next combination, let us see. The next combination, here you can see that there is capital R and small y. Capital R and small y means that capital R round seeds, small y means green. Okay. So here a different combination has come, round and green seeds. So how many round and green seeds are there? Let us see. 1, 2 and 3. So there are 3 round and green seeds. Now let us see the next new combination. You can see wrinkled and yellow, that means small r and capital Y, capital Y. Capital Y means it will be yellow, small r means it will be wrinkled. As the 2 small r's are there, that means it is a recessive trait in homozygous condition and it will be expressing itself. So how many combinations of wrinkled and yellow seeds are there? 1, 2 and 3. So 3 combinations of wrinkled and yellow seeds. Now only one thing or one combination is there, that is small r, small y and small y. So that is wrinkled and green. Only one combination of wrinkled and green seeds. So what is the phenotypic ratio we have got in this dihybrid cross? Let us see. It is 9:3:3:1. We are getting 9 round and yellow seeds. We are getting 3 round and green seeds, and again we are getting 3 wrinkled and yellow seeds, and only 1, that is wrinkled and green seeds. So in case of a dihybrid cross, the phenotypic ratio is always 9:3:3:1. So this was about the dihybrid cross. So in dihybrid cross we get a ratio that is 9:3:3:1. So we have seen the Mendels monohybrid cross and Mendels dihybrid cross. Now let us move to the laws which have been derived or which have been postulated on the basis of this monohybrid and dihybrid cross. So we have seen the Mendels monohybrid and dihybrid crosses. We have seen the ratios in mono and dihybrid crosses. Now on the basis of the observations by Mendel, he have given few postulates. He framed few postulates which later were framed as Mendels Laws of Inheritance. On the basis of Mendels monohybrid cross, 2 laws were given. Number 1 is the law of dominance and number 2 is law of segregation and purity of gametes. Mendel have given 3 laws, but on the basis of monohybrid cross, 2 laws are there, law of dominance and law of segregation or purity of characters. Now let us see what are these laws. Number 1 is law of dominance. Let's see the statement, which is very much familiar statement. In a heterozygous condition, the allele which is able to express itself phenotypically is the dominant allele, and the other allele which is not able to express itself phenotypically but is present genotypically, that is the recessive allele. So this dominance and recessive concept is applicable only in heterozygous condition. As we have seen in monohybrid cross, when we have taken homozygous tall and homozygous dwarf plant, when they have combined, we have got heterozygous tall plant. So why it is heterozygous tall plant? Because the dominant allele, that is the tall allele, is able to express itself and suppress the dwarfness allele. So the allele which expresses itself is the dominant allele, and the other allele which is being suppressed by the dominant allele is called as the recessive allele. The recessive allele is present genotypically, but it is not able to express itself phenotypically. So this is the law of dominance. Now moving to the law of segregation and purity of gametes. Now, law of segregation or purity of gametes states that alleles of a pair representing a character segregate such that the gamete receives only one of the two alleles and they do not show blending. Both traits are seen in the F2 generation. Now let's again go back to the monohybrid cross. You have seen that in F1 generation you have got all the tall plants, but in F2 generation you have seen the ratio was 3:1. 3 tall plants and 1 dwarf plant. So here the recessive trait is able to express itself in the F2 generation, which was suppressed in the F1 generation. Okay, so how this happens? You know, when the two alleles they come together, they simply associate themselves without mixing or blending. As you have seen in none of the crosses, we have seen intermediate plants. Why? Because the two alleles they simply come together, associate themselves at the time of hybrid formation. And again at the time of gamete formation, the two alleles they separate or segregate from each other, or we can say that they simply dissociate from each other. Okay, so here there is no mixing or no blending of alleles and the characters. Each allele is able to express itself. In the F1 generation, the dominant allele has expressed itself, while in the F2 generation, the dominant as well as the recessive allele was able to express themselves. So, we have seen in the F2 generation, both the traits are able to express themselves. 3:1 was the ratio, that means 3 dominant allele was there, like 3 tall plants were there, and 1 recessive trait was there to express itself, that is 1 dwarf plant was there. So, the character which was not able to express itself in the F1 generation has expressed itself in the F2 generation. So, this was about the law of segregation, that means the alleles they separate or segregate at the time of gamete formation, and again they associate themselves at the time of hybrid formation. So, the same is the purity of gametes, that means the gametes remain pure whenever the hybrids they form the gametes, they simply dissociate from each other. So, that is the law of purity of gametes. So, these two laws, law of dominance and law of segregation or purity of gametes, was on the basis of observations of the monohybrid cross. Now, on the basis of observations and results of the dihybrid cross, the third law of inheritance was given, that is the law of independent assortment. This law was given on the basis of dihybrid cross. Now, this law of independent assortment is somewhat similar to the law of segregation. In law of segregation, we have considered one gene and two alleles. But in dihybrid cross, you remember that we have considered 2 genes and 4 alleles, and it is also applicable for the polyhybrid crosses, where we are considering number of genes and their number of alleles. So, this law is for the dihybrid and the polyhybrid crosses, means this law has been derived from the di and polyhybrid crosses. So, let us see what is this law of independent assortment. The factors or alleles controlling different characters assort independently without influencing each other. Like in case of law of segregation, 2 alleles were there, and these 2 alleles, they were not mixing or blending with each other. They are simply associating and dissociating. The same thing is there in case of number of genes, as the chromosomes consist of number of genes and their number of alleles are also there. So, whenever these genes, they are transmitted to the next generation, or we can say whenever these alleles, they are transmitted to the next generation, they do not interfere with each other, they do not influence each other, like we have seen the combination in dihybrid cross, like there was yellow seed and green seed. So, whatever combinations we have got, that is either yellow, either green. We have not got a mixed color, that is a color intermediate between yellow and green. So, it means that all the alleles, they will assort independently themselves, or we can say that all alleles assort independently and they do not influence each other, and without influencing they pass from one generation to the next generation. So, this law of segregation is for 1 gene and 2 alleles, while this law of independent assortment is for 2 or more than 2 genes and its alleles. So, whether the genes or alleles, while moving from one generation to the next generation, they do not influence each other, they present themselves independently in each of the generation. So, in this class, we have studied about the Mendels dihybrid cross, the Punnett square, and all the 3 laws of inheritance, in which the Mendels law of dominance and law of segregation was the result of monohybrid cross, and on the basis of dihybrid cross, the law of independent assortment was given. So, we have come to the end of this video. I hope the Mendelian genetics is clear to you. Thank you and take care.
Thanks for watching. If you like this video, make sure to like, share and subscribe for more.